I'm Madelaine Frazenburg, presenting basic principles of biological sample preparation for scanning electron microscope. Part of the electron microscope unit, Central Analytical Facility, Stellenbosch University. Why is sample preparation important? Conventional SEMs operate at very high vacuums to avoid gas molecules interfering with primary and secondary backscattered electrons emitted from the sample. Everything going into the SEM must be completely dry and free of organic contaminants that may potentially outgas in a high vacuum environment. This poses a problem when dealing with biological samples, which are largely composed of water. To retain the native structure of an organism, requiring additional preparation steps. These steps are necessary depending on the type of sample and the purpose of the study. Some types of biological sample, for instance insects with hard exoskeletons and woody plant material, will require less stringent processing to preserve their structure, whereby the more delicate types of samples will take more time and care to prepare. Why is sample preparation important for EM? Sample preparation are essential because flawed samples preparation can undermine the quality of results and lead to false conclusion. It is necessary to avoid the introduction of drying artifacts like sinkage and collapsing. Artifacts can be undesirable things to be avoided if they do comply with preconception. It is important to compare images obtained by different technical methods, for example, transmission electron microscope with the light microscope or scanning electron microscope, and under different conditions from which we can subtract artifacts which we have been able to distinguish. No single step during the preparation of biological material for viewing can be regarded as independent of each other. An analysis of each separate step is necessary for some tentative understanding of its contribution to the final result. So look at the picture of Bacillus cereus cells in sample A or image A were washed with distilled water and oven dried at 40 degrees. The cells are flattened, shrunken, and covered with dried mucus layer. In image B, we see that the Bacillus cereus cells were air-dried using HMDS protocol. The cells appear rounded, well-exposed dividing cells. Primary consideration in sample preparation for SEM. Obtaining acceptable image with good ultrastructure preservation requires careful application of the SEM sample preparation method. Making an effort to do some research and preparation prior to collecting the samples. Consider journal articles in which authors prepared the same or similar type of samples for SEM. Always use a sharp blade to avoid deformation. Make sure you know the best fixative to use and that you have prepared and ready to go when you collect your samples. Transfer your samples immediately into the fixative to halt any cellular changes in the organism after you harvest or killed it, to avoid autolysis, putrefaction, and drying artifacts. Aim to collect and fix something that is small as possible to ensure good infiltration of the fixative into the sample. You will need to dissect an area of interest of your sample prior to fixation. Once the sample has been placed in a vial with primary fixative, the sample vial should be used throughout the sample preparation protocol. Do a pilot study using 1 or 2 samples to determine the correct sample preparation for your type of sample. Fixing agents. There is different types of reagents that can be used to fix tissues. Like formaldehyde, by far the most popular reagent used for histopathological, and glutaraldehyde, widely used for ultrastructure studies requiring electron microscopy. Formaldehyde is the only gaseous aldehyde and is dissolved in water to saturation at 37 to 40%. Paraformaldehyde, a highly polymerized form of formaldehyde, may be deposited as a white precipitate in concentrated formaldehyde solutions. To prevent this, Small quantities of methanol, up to 15%, are commonly added to proprietary, patented or branded solutions. Unbuffered formalin will slowly oxidize to form formic acid, resulting in a fall in pH. Formic acid will react with hemoglobin, forming an acid formaldehyde hematin, a brown-black granular artifact. A pigment is deposited in the blood-rich tissues. This pigment is a nuisance as it can be confused with microorganisms or other pathological pigments. Formaldehyde reacts most effectively at about a neutral pH. 10% formalin solutions are usually buffered to pH of 6.8 to 7.2. In sample A, we see sample from a culture. Fixative must be added directly into culture plates. Fresh microbial cultures gave better results. Don't leave cultures in the refrigerator for long periods of time. Glutaraldehyde, another fixative, is one regarded as the bifunctional aldehyde, possessing aldehyde groups at either end of the molecules which have the potential to react with same chemical groups as formaldehyde. Tissue fixed in glutaraldehyde will be more extensively cross-linked than tissues fixed in formalin, and will also possess some unreacted aldehyde groups that, unless chemically blocked, can cause background staining in methods such as PAS. The extensive cross-linking adversely affects immunohistochemical staining, but does provide excellent ultrastructural preservation, which explains its extensive use as a primary fixative for electron microscopy. Cross-linking reaction of glutaraldehyde are largely irreversible. Glutaraldehyde penetrates very slowly, and it is recommended that tissues be less than 1 mm in thickness in at least one dimension. Glutaraldehyde can be added to a suitable buffer at a pH of 7.2 to 7.4, usually cacodylide, to produce a 3% glutaraldehyde concentration for use. For electron microscopy, glutaraldehyde primary fixation is commonly followed by a secondary fixation in osmium tetroxide. Factors that will influence the rate and effectiveness of a sample fixation. First, temperature. Increasing the temperature of fixation will increase the rate of diffusion of the fixative into the tissue and speed up the rate of chemical reaction between the fixative and the tissue elements. It can also potentially increase the rate of tissue degradation in unfixed areas of the specimen. It is believed that fixation at 0 to 4 degrees reduces the possibility of the extraction of cytoplasmic elements from cells. Secondly, time. The optimal time for fixation will vary between fixatives. For fixation to occur, the fixative has to penetrate by diffusion to the center of the specimen, and then sufficient time has to be allowed for the reaction of fixation to occur. Both diffusion time, reaction time depends on the particular reagent used, and the optimum time will vary from fixative to fixative. Another factor is specimen dimension. A specimen should not be more than 4 mm thick. Ideally, a 3 mm thick slice should provide excellent fixation and processing. It is useful to remember that the specimen cavity in a standard processing cassette is 5 mm deep. Volume ratio. It is important to have an excess volume of fixative in relation to the total volume of the tissue, because with additive fixatives, the effective concentration of reagents is depleted as fixation proceeds, and in a small total volume, this could have an effect on fixation quality. A fixative tissue ratio of 20:1 is considered the lowest acceptable ratio, but I would recommend a target ratio of 50:1. The next factor is your penetration rate. The penetration rate of a fixing agent depends on diffusion characteristics and it varies from agent to agent. Osmolality generally is the osmolality of a vehicle that is most important, and in some formulation this is adjusted to resemble that of tissue fluid, example formalin in isotonic saline. Before fixation occurs, cells can certainly be damaged by non-isotopic fluids, such as water, and if specimen cannot be immediately fixed, they can be kept moist with a gauze soaked in isotonic saline for a short period of time. It is not advisable to hold tissues immersed in saline for extended periods. For marine samples, osmolality is achieved by using seawater instead of buffers. Hypertonic solutions give rise to cell shrinkage, while hypotonic solutions result in a cell swelling and poor fixation. pH and buffers does not appear to affect the quality of preservation greatly, as a number of formulations have quite a low pH, such as those containing acetic or puric acid. However, pH can be important for other reasons, as in case of formaldehyde solutions, where breakdown of formaldehyde to form formic acid produces an acidic solution, which in turn reacts with hemoglobin to produce an artifact pigment. The most popular formaldehyde solution today is therefore buffered with a pH of 6.8 to 7.2 for this reason. In our picture, we see that freshwater and marine water samples like diatoms can be fixed in routine fixatives. However, marine samples, the fixative should be prepared in filtered or artificial seawater with its osmolality matching their natural environment. Samples with heavy mucus films obstruct the clarity of the ultrastructure. Mucus layers can be removed by washing of the sample with suitable saline before fixation. In the image A and B, we see the removal of the mucus layer in these intestinal parasites avoids stress in samples. Small organisms like worms, water insects, zooplankton tend to show some form of stress or even struggle when immersed into the fixative. These stresses effects may result in change or loss of ultrastructural components of the sample. It is advisable to narcosize or slow down the organism before fixation. Leave the sample in the fridge for about 5 to 8 degrees for a short period of time before fixation, or immerse the sample in magnesium chloride solution or dilute alcohol before fixation. In the image, we can see a mite that has been collapsed during the sample preparation protocol. Samples. The samples must need the size that can fit into a chamber. The sample must be dry. You can either use a critical dryer or freeze drying or drying reagent like HMDS. It's important that the sample is conductive, so you can use the sputter coat and coat your sample with a heavy metal, for example with gold, palladium, platinum, iridium for high resolution images, or carbon for qualitative analysis. Sample preparation methods. There's different types of methods how to prepare the sample. So the sample can be transferred into a small multi-wall plate, microporous capsules, or custom-made chamber. Continue to fix with paraformaldehyde and glutaraldehyde. Never allow a sample to air dry. Samples should remain submerged in fixative. Keep cold at 4 degrees. Left in fixative for longer than an hour. Sample collection, getting started. Collection of samples for SEM preparation in a laboratory involves the dissection of an animal or incision of a plant material to remove tissue from the main body. All incisions must be performed with fresh blades to avoid deformation of tissues from the undue physical forces needed with a blunt blade. Use the same vial throughout the protocol. Simply decant or pipette out the changes to minimize handling. Never freeze specimen. Fix specimens in aldehydes, for example 4% formaldehyde and 2% glutaraldehyde, to cross-link proteins and keep them cold at a 4-degree temperature for prolonged times until post-fixation and further processing for SEM analysis can happen. Post-fixation with osmium tetroxide. Osmium tetroxide fixes lipids and therefore stabilizes membranes and ultrastructural features. Osmium provides conductivity under the scanning electron microscope. Osmium is a heavy metal that enhances the contrast, therefore provides a stronger electron signal and can be obtained with soft biological material containing mainly carbon, hydrogen, oxygen, nitrogen components. Dehydration step. Dehydration is done in graded series of ethanol to remove water gradually and gently, typically with percentages starting from 30%, 50, 70, 90, 100%, with 10 to 15 minutes in each concentration step. Repeat the final 100% ethanol step to make sure that all residual water has been removed. Final drying process. Final drying after 100% ethanol can be done in various ways. Number 1, you can try with critical point drying option with liquid CO2 in this critical point drying apparatus. Or you can use the chemical drying option with HMDS or freeze drying option under the vacuum using liquid nitrogen. Here I present a flow chart of air drying A with HMDS and critical point drying with liquid CO2. And critical point drying with liquid CO2 sample preparation methods. Both methods are known to be equally effective in ensuring good preservation of the structural integrity by minimizing or eliminating the impact of surface tension effects in the sample. Sample mounting and conductive coating. Mount dried samples on aluminium SEM stub using a brush or thin needles. The SEM stub is covered by conductive carbon tape or carbon paint or electrodag. Sputter coat a sample to enhance conductivity, typically with gold or gold palladium or any other metal or carbon. Biological samples being non-conductive give rise to charging problems in the SEM, as the bulk or primary electrons from the electron beam remain in the sample to form clouds of negative charges. This charge build-up interferes with the primary beam to bring about image distortions, loss of contrast with very bright and dark areas, known as charging effect. To overcome these problems, a thin layer of metal is sputtered on the sample, thereby increasing the conductance on the sample to enable the absorbed electrons to find their way to ground. A sputter coater is used for this purpose. The referred metal for sputter coat are gold, gold palladium, platinum, and chromium. However, platinum and chromium are the choice metal for high-resolution images, more than 50,000 magnification, as gold sputter result in the visible graininess on the surface of the sample. Particular samples are dry powders, fine crystals, nanoparticles, dried bacterial cells, and spores, and they do not need any special sample preparation protocols. Care must be taken to avoid loosely stacking one over the other. Particles must be firmly stuck on the specimen stub. Stacking of particles causes instability and charging problems. You can follow the following procedure. Sprinkle a little of the sample evenly but lightly on the SEM stub with double-sided carbon tape. Use a hand blower to blow away the loose particles. Always blow away from yourself into a bin or sink. Employ increased safety procedures if the particles are known to be harmful. The sample is now ready for viewing in the SEM. Sputter with either gold, platinum or chromium. There's different options how to load the sample in the chamber. Firstly, you get a chamber loading zone that you don't break the vacuum in the big chamber, or you have SEMs like the EVO SEM where we have to break the chamber vacuum and flush the chamber with nitrogen to load the sample. Getting started by using SAF-EM unit, we can see our contact details and we're looking forward to hearing from you. Thank you very much.